The discussion of white blood cells that we finished the last section on is a great way to get into now the immune system. As we introduce the immune system, now instead of talking about pathogens and viruses, now we're specifically going to focus on an antigen, which again can be a pathogen, it can be a foreign material, but the specific term for antigen means that it's something that can cause antibodies to be generated. And that's where we get the term antigen. It can also be referred to as immunogen, something that generates an immune response. And as we look at our body, it's geared in many ways to recognize foreign substances. And in this illustration, we have 4 portions of a pathogen, maybe a bacteria. And each orange portion refers to a specific epitope, a specific portion that can be recognized by an antibody. And these are the proteins we see in blue that have this Y shape. And we're going to get into the antibody structure by the end of this lecture. And you can see that these antibodies have the capacity to bind to an antigen. And in this case, this antibody and its curved recognition site on the end of the arms, it's going to recognize this particular pathogen. Is there another pathogen that this antibody will be able to recognize? Yeah, not just the one on the left, but over here on the right, because it also has that same antigen. So these small antigens on these different bacteria, that's what's recognized by these parts of the immune system. And when we talk about antibodies, we're talking about B lymphocyte generated and humoral immunity. But before we get into the specifics of it, your body also has a whole series of mechanisms that can prevent infections, resist foreign materials in non-specific ways. These defenses don't care what the pathogen is, nothing's going to get in. And so we refer to these collectively as the innate or non-specific immunity. Great, it's non-specific, it's going to be fairly robust, but there's really no memory, no long-lasting effect that we see with our antibodies and our specific immune system. But this takes care of most of what we need to prevent you from getting an infection. And in fact, one of our best defenses in the innate immune system is your skin. Why is your skin so good at preventing an infection? Or let me put it this way. When do you get an infection in your skin? When you get a cut, when you compromise the integrity of the skin, when it's no longer contiguous or all together. So just the contiguous nature of skin, it's a physical barrier. Your skin is also fairly alkaline, so it's not a real pleasant environment for a lot of pathogens. Plus, your skin is continually producing new cells at the bottom, and the cells at the top are being shed, desquamated. We learned that term in a lecture or two ago. So basically, pathogens are swimming upstream to try to get into the deep parts of your body. So the skin is a great barrier. So if a pathogen can't get through your skin, another way to get in is through your mouth, the digestive tract. And what's the very first thing that a pathogen is going to swim in when it hits your stomach? Hydrochloric acid. Is that going to be very fun? No. So you can't get in through the skin. You can't get in through your digestive tract. What about respiratory tract? You can breathe in stuff all the time. Well, we have cells that line your respiratory tract that secrete mucus, the sticky, nasty material. So your pathogens get stuck in the mucus. We also have cells that line your airway that have cilia. These cilia beat upward. So the pathogens get stuck in the mucus. The cilia move the pathogens in the mucus up to your pharynx, larynx. And then what do? Swallow it into a pool of hydrochloric acid. So there are a number, and does it matter what that pathogen is? If it's a virus, if it's a bacteria, if it's a parasite? No. It's going to do the same thing regardless. Non-specific protection. So when we look at the innate immune system and the cells of the innate immune system, the macrophages, the granulocytes, they're phagocytic. If it doesn't belong, they're going to eat it. So we see that in a non-specific way, these immune system cells are going to specialize in not only the removal of some of these pathogens, but also in the production of these chemical signalers called cytokines. Let's say you happen to go into one of your favorite stores and you see something on sale, just redonkulously cheap. Are you going to go buy this thing and go home and wear it, play with it, and never tell a soul? You're going to be texting, you're going to be tweeting, you're going to be Insta-chat-gramming or whatever you do to get the word out. You're going to be telling everybody. So these non-specific responses in the immune system, when cells recognize a pathogen, they're going to start calling all hands on deck with these chemical signals that say, hey, I don't know what it is, but it doesn't belong. Come help me get rid of it. And that's what these chemical signalers called cytokines. They're very adept at producing these chemical signals and following them to the point of the infection. Now, our white blood cells are also very good, along with our antibodies, again, in a non-specific way, of activating what is called the complement cascade. Has anyone heard of the complement cascade? That's one of these immune system things that I never had heard about until I took this class a long time ago. These are proteins that exist in the plasma, much like our clotting factors. They're in an inactive state, but once they become activated, they can lead to an enhancement in many ways of inflammatory responses, antibody-mediated responses, as well as increasing the phagocytic activity of our nonspecific white blood cells. So it ramps up the immune system in, again, a non-specific way. Macrophages, neutrophils. They know when foreign material is present, but they don't really care if it's a specific one or the other. Macrophages is going to go, I can't eat that. That doesn't agree with me. No, I would rather if that was chicken and not beef. No, macrophages are just going to eat. Eat. So they're going to identify foreign material and remove it from the body. If it doesn't belong, it's gone. Period. Now, many of these through cytokines and through other receptors are going to be able to activate the other branch of our immune system, the adaptive, the specific. For instance, macrophages. They eat everything, right? Macrophages are also a category of cell we call antigen presenting cells, or APCs. Macrophages, if it's foreign, they eat it. But then macrophages do this really gross thing. Have you ever been eating something and something gets stuck in your tooth and you gotta get it out? There's nothing worse than either that popcorn hull, or have you ever got the nacho chip stuck? You just got to come out. Well, macrophages get this foreign material, they eat it, and they pick out of their teeth and they go, ooh, what is that? And so they start crawling around in the body asking other cells of the immune system, do you know what this is? Do you know what this is? Until it finds, that was funny, right? Until it finds the white blood cell that goes, yeah, I know what that is. That's chickenpox virus. And then the macrophage goes, hey, follow me. I'll show you where it is. And it recruits those specific immune system cells that can get rid of that specific pathogen. Even though the macrophage didn't know what it was. He just ate it. And there's our macrophages. Again, because macrophages eat anything, well, when they sense phosphatidylserine, which is a portion of human and mammalian and animal plasma membranes, they know that a cell somewhere has undergone apoptosis, programmed cell death. And so the macrophage comes in and goes, as one of my buddies, he kicked the bucket and eats what's left, cannibalizes that particular cell. I'm telling you, macrophages eat anything. Kind of like my boys at home. Now, this is an interesting chart. We're not going to learn all of these pathways, but what I want to show you is you have receptors on the surface of cell, the plasma membrane. And when you get materials binding to these receptors, look at the cytoplasmic face. You see this FADD. That's a cytoplasmic portion, and the DD stands for death domain. That starts a signaling cascade, which leads to the activation of caspase enzymes. The caspase enzymes make their way into the nucleus and shred the DNA. And that's one way in which cells undergo apoptosis, by the activation of the death domain receptor. Mitochondria start breaking down. They release cytochrome C. You can also have P53 released. All of these are signals that, okay, cell, there's something wrong. We need to kill our cells before we make it worse for the others. And once they're gone, the macrophages come in and clean it up. Now, in our discussion of white blood cells, we talked about basophils. And when they move into the tissues, we call them mast cells. These large blue granules that, among other things, contain heparin and histamine that lead to inflammation. Now, inflammation, we often think it's a bad thing, but actually inflammation is part of the immune response. And during the course of inflammation, there are 4, possibly 5 signs of an inflammatory response. Sometimes they're called the 4 ors, for the old Latin and Greek: rubor, redness, tumor, swelling, calor, heat, and dolor, pain. But what is the redness? When you have something swollen, something inflamed, why is it red? Increased blood flow, which again is also going to be for calor, increased heat. More of your core body temperature is brought to the region because of the blood. We have this swelling, this puffiness. Clinically, we refer to it as edema, because what does the heparin make your blood vessels? Makes them more leaky. Makes them more leaky. And so that's the tumor, the swelling. And because you're putting more pressure on tissue that's not accustomed to that kind of pressure, you get the pain. And if you have enough edema, you can actually get to this 5th one, which is loss of function. Now, if you're in a clinical setting and you see one that has so much edema they've lost function, that's a bad thing. But you never know what you're going to see in a clinical setting. It's amazing what people will endure before they go to the doctor. Either because they don't trust the doctors, don't like the doctors, don't have insurance, don't have the money. It's absolutely amazing. So here we see an illustration. Bacteria have come through a cut in our skin. The bacteria are going to be recognized by antibodies, which we'll talk about in just a bit. And then we have our macrophages that gobble up this material and they go, oh, wait a minute, what is this thing? And you can see the blue signals that we have. And here's our mast cell that's activated. It's releasing its heparin and histamine, increased blood flow, increased permeability of the blood vessel itself. Whoop, here comes another phagocytic type cell, probably another macrophage because the neutrophils may already be here. And it's coming out of the circulation, goes, oh, where's the party? Where do I eat? And the mast cell is part of what's helping signal and get those leukocytes to the scene of the infection. Now, even though we mentioned antibodies here, antibodies are part of the specific immune system. However, the specific part is what is recognized by the antibodies. Antibodies themselves that have decorated a pathogen are going to non-specifically increase the phagocytic activity of macrophages, and that process is called opsonization. If you've been in any of my classes before, you know I love chocolate. I love chocolate. You could put chocolate on a rock and I'd probably try to eat it. I love chocolate. Well, antibodies on the surface of a bacterium To macrophages, which is opsonization, for me, that would be like chocolatification, if you put chocolate on the bacterium. So these antibodies just make that pathogen extra yummy to the macrophages. It doesn't matter what the pathogen is. It doesn't matter what type of antibody is there. Macrophages see extra antibodies. They're like, I want 3 of these. Keep them coming. And they phagocytose them much more rapidly. So mast cells, we've already talked about an inflammatory response. Mast cells, heparin anticoagulant, histamine, vasodilation. But we also see in those granules of the basophil/mast cell, we also have prostaglandins. Prostaglandins lead to pain and fever. We don't like either one of those. But pain is extremely important. Pain tells you, stop doing what you're doing because it hurts, or you need to fix whatever's going on because there's something bad happening. Fever isn't a bad thing either. Fever is trying to create an inhospitable environment for the pathogen. And yet as soon as we have any kind of fever whatsoever, I don't know about you, but I am taking some ibuprofen or Tylenol or something. I don't like fever, period. Now maybe that's a guy thing. You ladies, I know y'all are a lot tougher than we are. Moms can have 150 degree temperature and they're still taking care of 3 kids, making dinner, doing laundry, Dad, 98.7, call 911. I'm in bed. I don't know. Leukotrienes are also present in our mast cells. Again, increasing vascular permeability, making that blood vessel leaky, making it easier for the white blood cells to diapedese out of the circulation and get to the tissues where you typically have the infection. That's what all these things are geared toward. So many of these cytokines from the mast cells, they're going to be assisting in getting more blood there and getting the cells out into the tissues. We've already touched on neutrophils, macrophages. They're going to come in and gobble up neutrophils quick. The macrophages coming in a little slower, but both work together to clear the pathogen out of the area. And what is left over? Pus. Yeah. Now back to fever just a little bit. This inhospitable environment, it doesn't feel nice for us, and I can't imagine just from a logical sense how the cells of my body like it since we're so incredibly hot. But when we look at fever and in fact what fever is trying to accomplish, not only is it inhospitable, but for our leukocytes, it speeds them up. They move faster. They eat faster. And for bacteria that produce these endotoxins that can cause tissue damage, that increased temperature actually decreases those effects of the endotoxins. So it's not just making it uncomfortable for the bacteria so the bacteria die. Man, there's a lot of other things going on. And in fact, T cells, they divide faster. These are part of your cell-mediated immune system. And in fact, fever is also going to enhance the activity of interferon, which we're going to look at on the next slide. But here's a question I want to pose to you. When you have a high fever, 102 degree fever, what sort of physiological response do you usually experience when you have a high fever in much the same way you experience when you're very cold? Chills. You get chills. When you're outside on a cold day, you shiver. What are you trying to do? Warm your body up. That's because your thin actin and your thick myosin are sliding back and forth across each other, creating friction, generating heat. But when you have 102 fever, you're burning up. Why are you shivering? Why do you have the chills in the same way as you do when you're outside in 20 degrees without a jacket? Any clue? Ever wondered that? So right now, in this room, our ambient temperature, I don't know, what we say, about 72 degrees? And some people are cold, some people are comfortable, some people might be warm. Okay, that's sort of the acclimation to the temperature. But our feeling of the temperature, this is the temperature in the room. Our body temperature, 98.6, is here. Now, if we're cold, what do we usually do? Go to the thermostat and do what? Turn the heat up. So what we want to do, if we're cold, we want to heat the ambient temperature and move that temperature up and decrease the difference between room temperature and our body temperature. And if we're hot, we go turn the AC on, make that difference bigger. When we're outside and it's cold, our body temperature of 98.6, that difference is bigger, right? So we're cold, like, I got to warm up. But now if we keep the room temperature the same and we're sick and have a fever, we increase the difference because of the fever, but it's not communicating with the other part of our brain to go, no, stupid, we're not freezing to death, we have a fever. But you start to shiver because that difference between your body temperature and ambient temperature is bigger than normal. And so you shiver. And then as soon as your fever breaks and it goes back down, because when you got the shivers and the chills, what do? You put on 2 pair of socks, your sweats, 10 blankets, and you go, okay, that feels better. You increase the ambient temperature around your body. Your fever breaks now, what are you doing? Man, you're throwing covers and you're kicking things off because you're starting to sweat. Your fever did what? Fever broke. That's how you can tell when that's going on. So, we said fever, among other things, enhances the activity of interferon. This is another one of those cool words, antigen, antibody generating substance. Interferon is a substance produced by your leukocyte that interferes with viral replication. That's how viruses replicate. They get in your cell, turn the cell into a viral factory until the cell dies, releases all these viruses so they can do the same thing. This chemical released goes in and it inhibits viral replication. But interferon also activates your killer T cells and macrophages to say, hey, you need to go get rid of that cell's got all these viruses in it. Before it releases them, they go everywhere. Man, a ton of research has gone into interferon to enhance chemotherapy kinds of drugs. I don't know how far it is advanced. I've seen some, read some papers, seen some documentaries on it. I don't think they've really arrived at the actual right place to use interferon effectively. But it would be great if we could enhance your body's ability to produce interferon and to take care of viral replication. So that was the innate, non-specific. Now the adaptive, specific immune system, this is still going to be able to recognize foreign material, but it's going to do so in a very specific way. And this system is going to have a memory. It's going to be able to remember, hey, I saw you before, you don't belong here, I'm going to get rid of you. This is going to be the foundation with which we do our vaccines and vaccinations. That prevents us actually getting a specific disease. So this is an illustration that puts the innate on the top together with our adaptive immune system. So in our illustration, we have this dendritic cell, which is going to be an antigen-presenting cell that is then going to be able to stimulate macrophages, stimulate inflammation. There's interferon. But it can also signal and start the process of activating cells in the adaptive, the specific immune system. And again, what are those cells? They're the lymphocytes, the B's and the T's. So our lymphocytes are going to have their stem cells, again, in the bone marrow. But as these cells develop and before they mature, they leave the bone marrow, get into circulation, and end up in a lymphoid tissue where they become capable of recognizing pathogens. And our two types are the B and the T. B lymphocytes become immunocompetent in the bone marrow, hence the B. And it's these cells that will, when exposed to antigen via receptors, begin to divide, and they're going to produce cells called plasma cells that are antibody factories. And these antibodies are going to be made, and antibodies are going to bind to one specific antigen. Each plasma cell is going to produce its own set of antibodies. Versus our T lymphocytes that mature in the thymus, and that's where the T comes from. These T lymphocytes are going to basically be cell mediated. They're going to directly bind to a cell and instruct it to be destroyed, or they're going to secrete materials near that cell that's going to kill it. So again, back to our whole military comparison here. Neutrophils were the who? Who were the neutrophils first in? They're the Marines. Macrophages were the reinforcements. Well, now the B lymphocytes, they're the Air Force. They're dropping the bombs. The antibodies are the bombs at a distance. The T lymphocytes, this is the foot soldier, the infantry. They've got the bayonets. They're doing hand-to-hand combat or throwing hand grenades. That's the soldiers that we have in our immune system, the white blood cells. So this illustration shows our common bone marrow stem cell, eventually giving rise to the distinction between the B lymphocyte and the T's. The T lymphocyte is either going to produce a helper T, regulatory T, or a killer T. The B lymphocytes, as they get exposed to antigens and they clone themselves over and over, make hundreds and hundreds of copies, they're going to eventually produce a plasma cell that's our antibody factory. But they also can produce memory cells, which are the cells that are inactive and just kind of waiting for a subsequent exposure somewhere down the line. Maybe next week, maybe next month, maybe 30, 40 years later. That's what gives us the ability to remember a pathogen that's been seen before. Now, our B lymphocytes, again, they start cloning themselves after about 5 days, and when they first recognize this pathogen, they're going to create plasma cells that produce antibodies, maybe after about 2 weeks. But during this process of cloning, they also produce these memory cells that when memory cells are subsequently exposed to that same antigen, they start producing antibodies in 2 hours, not 2 weeks. There's a whole lot more memory cells, and they're much more easily activated to produce antibodies, so you have a much more rapid and a much more robust response to an antigen than you do with simply our B cells in the first exposure. So we just have an illustration showing B cells with their antibodies, which are on their surface, that are their receptors. When the antigens bind, that turns on the mitotic activity. Cloning, 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 cloning. Plasma cells, and also memory cells. This illustration shows you it takes about 5 days before we really see any antibody of any consequence appearing in the blood. And yet at the same time, we're developing the memory cells. Now, they're not showing the entirety of this pathway because as these cells divide, you're seeing an exponential expansion. You start with 1, and then as you go through the first division, you have 2, then 4, then 8, then you see what I'm saying? That's how you can get so many more cells present after you specifically activate the first one. And all of this lineage is, for all intents and purposes, going to recognize the same antigen. Now, early on, we said epinephrine is not a neurotransmitter. It is a what? Hormone. B cells do not make antibodies. You're going to see this in textbooks. You're going to see this in papers. You're going to hear me say it, but correct me if I do. B cells don't make antibodies. B cells produce plasma cells that make antibodies. Now, you can't eventually get antibodies without the B cells, so I'm being a little ticky-tacky here, but I want you to be precise. B cells, when they start dividing, become plasma cells, and it's the plasma cells that are the antibody factories that can produce maybe upwards of 3,000 antibodies, which are proteins, per second. And antibodies being proteins, it's no wonder when you look in the cytoplasm, you see tons and tons of endoplasmic reticulum, rough endoplasmic reticulum, because that's the protein factories for secreted proteins. So antibodies are produced not by B cells. They're produced by plasma cells that are derived from the bees. So they're in the bee lineage. Now, we've talked about B cells, plasma cells, antibodies. Let's look at antibodies, these proteins that are important in this humoral immunity. Remember, we touched on them when we talked about the proteins in plasma. We call them the gamma globulins, immunoglobulins, antibodies. These are, in fact, going to be free-floating proteins in your plasma, but you may find them on the cell surface of B cells as that B cell receptor. Because here's the thing, that B cell is going to make one specific kind of antibody that recognizes one specific antigen. How is that cell going to know that that antigen is around if it doesn't have that receptor to bind to it? I look at it like it's the label on the aluminum can. You go to your pantry. How do you know what's inside that can? There's a picture or words that tells you and reflects what's inside the can. Well, this receptor on the surface is, in fact, what's inside the B cell. And if it's capable of binding antigen, then that cell's capable of making more cells that can recognize that particular antigen. So the B cell receptor, yeah, it's the antibody label of what that cell can do. Now, we saw that initial picture of that Y-shaped molecule. That is our antibody. It is Y-shaped, and it consists of 4 protein chains that are held together in one molecule through disulfide bonds. And so our antibody is going to have 2 heavy chains and 2 light chains. And when we put these together in this Y-shaped structure, we're going to be referring to 2 specific functional regions, one being the variable domain and one being the constant domain. And again, I use that term constant domain now for simplicity. We're going to change it up, and it's not quite as constant down the road. But we don't see as much variable in that part as we do in the variable region that actually recognizes antigen. So our picture, as we see in our illustration, on the left, we see the distinction between the heavy and the light chains. And again, where you see the dashed lines, these are some of our disulfide bonds. We have more than just 4, but that's all they've decided to illustrate here. Just letting you know they're there. And then from a functional distinction, on the right, we see in blue is what is considered the constant region, and the variable region is on the end. And again, I like the visual of like doing the YMC dance. So I'm standing here with my arms up. What do your hands do out here? What is their function, the function of your hands? You grab things. The variable region is where that antibody is going to bind antigen, in the variable region. And we're all humans. And so our legs are pretty much going to be very similar. And yet the actual shape and size of our hands is going to be fairly different from individual to individual. It's the same thing with antibodies. We're going to have several different types of antibodies that will define the constant fragment, and we're going to learn about 5 or 6. But there's an infinite variability to the variable region, and that's why we make the distinction that we do. So the variable region is where we're going to bind pathogen. If you notice, an antibody can bind 2 pathogens at the same time. So I want you to think of the antibody almost like handcuffs. You got 2 criminals, you handcuff them together. They're not as likely to just run away if they're both handcuffed together. Pathogen's not going to be very likely to get away when you start handcuffing them to each other and you make these humongous rafts where hundreds and thousands of pathogens are handcuffed together by hundreds and thousands of antibodies. It's these big rafts of antigens and antibodies that macrophages come in like sharks and just gobble up. And plus, handcuffing them together, the pathogens can't run away. So in this sort of space-filling model, we have the light chain in yellow, the heavy chain in blue. This is our variable region. So here we have a pathogen in green, and the red is the very specific antigen that's going to be recognized by that variable domain and have that perfect fit in that pocket so that this antibody can bind very specifically to this antigen of this pathogenic molecule. Now, the constant fragment, which is often abbreviated the FC fragment. These are going to be the legs, the stands of our antibody. And while the variable region might be able to bind to a lot of different pathogens, those constant fragments decorated and sticking off the pathogen, it's the FC fragments that drive macrophages crazy. That's the chocolate part. Not necessarily the variable part, the FC part. That's the part that opsonizes, that word we used. The FC fragment opsonizes that pathogen. Now, these next 2 ideas, you're going to see these more in basic science settings, but it's other ways in which we can define the antibody to use. Experimentally, if we don't want the constant fragment around to opsonize, maybe we're injecting stuff into experimental animals, or if we want this thing to bind to a molecule to either inactivate it or to localize it, but we don't want to handcuff 2 together, we can enzymatically go in and modify the antibody. And we can designate these either the FAB or the FAB2 fragment. And we can illustrate them here. Here's our constant fragment. And if we use enzymes just to cut off the ends, just to cut off the variable regions, we will have 2 independent molecules that can bind but can't cross-link materials together. And if we use a different enzyme and we just cut like the knees off, we still leave the cross-link molecules together, but the end of our constant fragment is the opsonization part. Then this part of the incomplete antibody we call the FAB2, really is FAB squared, and that's the fragment. You won't find FAB2s and FABs in nature. That's experimental materials that we use in the lab. You'll see these a lot in cell biological or physiological types of studies and papers. So I wanted you to be familiar with what these designations were as it relates to antibody structure.